Hello friends, in the last video we discussed about extranuclear inheritance or cytoplasmic inheritance. We compared nuclear genes and cytoplasmic genes, then discussed about the characteristics of cytoplasmic inheritance. We discussed about extranuclear inheritance by taking the example of experiment that was performed by Carl Correns on Mirabilis jalapa, that is 4 o'clock plant, and this involves the inheritance by chloroplast. Since the chloroplast still has its own DNA and it is present in the cytoplasm, it is also known as cytoplasmic inheritance. You can refer my video, the link is given in the description box. Today in this video we are going to discuss about extranuclear inheritance by mitochondria. Here we will discuss about the inheritance pattern of petite mutants. Like chloroplast, mitochondria also consist of its own DNA, and this mitochondria also present in cytoplasm, so it is also known as cytoplasmic inheritance. So what are these petite? Petite mutant. It was first discovered in yeast Saccharomyces cerevisiae, that is also known as the baker's yeast. In the yeast cells, normally what we see, large colonies when these are grown in the agar plate, and these are known as granae. But in rare cases, small or minute colonies are also seen. These are known as petite. This is due to the slow growth rate. Here the number of the cells are fewer compared to the cells present in the large colonies, but the size of the cell is same. And why these have the slow growth rate? Because this type of the cells consist of mitochondria which has defective electron transport protein. And how the mitochondria has defective electron protein? This is caused by the mutations in mitochondrial DNA. And since the mitochondrial DNA is present in mitochondria, which itself is present in cytoplasm, this type of the petite is known as cytoplasmic petite. There are also certain petites which results in the mutations in nuclear encoded genes, which are involved in the oxidative phosphorylation. Hence, these are not cytoplasmic petites. Inheritance of these petite was described by Boris Efrasi et al. What he observed, that there are three types of the petites: segregational petite, neutral petite, and suppressive petite. Let us see their inheritance pattern one by one. Inheritance of segregational petite. Here the petite cell, which is haploid, is crossed with the normal cell, which is wild type or grande. This is also haploid, and when these are crossed, this gives the diploid zygote, which is normal. Then the zygotic meiosis takes place, both meiosis 1 and meiosis 2, and we get the petite and normal ascospores in the ratio 2:2. So this ratio indicates that there is Mendelian segregation taking place. So what we can conclude, that segregational petite follow Mendelian segregation. So these are controlled by the nuclear genes, or in the segregational petite, the defective mitochondria is due to the mutation in the nuclear genes, because this nuclear genes only shows the Mendelian pattern of segregation. But there are certain petites that are also extrachromosomal or non-segregational. These are also known as the vegetative petites, and the genetic basis of these petites is the Rho factor. This Rho factor is the cytoplasmic factor, and when this Rho factor is absent or defective, then it results in the petites. So in this type of the extrachromosomal or cytoplasmic petites, or in the vegetative petites, this Rho factor is absent or defective. One such peptide is neutral peptide. It is shown by rho 0. That means it completely lacks the rho factor. Let us see its inheritance pattern when the haploid peptide is crossed with the haploid normal. Then it gets diploid zygote, and this is normal. Then this undergoes zygotic meiosis, and the progeny or the spores or the haploid cells which we get are all normal. So on the basis of this inheritance pattern, it was explained that neutral peptide lack most or all the mitochondrial DNA. So most of the genes are lacking, which encodes for the electron transport protein, which is involved in oxidative phosphorylation, and hence this is the cause of the defective mitochondria, which results in neutral peptide. So when this neutral peptide is crossed with the wild type, then the cytoplasm of the wild type is the source of the normal mitochondrial DNA in the resulting progeny spore, because cytoplasm of the wild type has the normal mitochondria, hence normal mitochondrial DNA. So the progeny which we get are all normal. Therefore, what we can say that neutral peptides are not transmitted. Now the next is suppressive peptide, which is represented by rho minus. This has the defective rho factor. Let us see its inheritance pattern when the haploid peptide is crossed with the haploid normal. We get the diploid zygote, which is peptide. And when this zygote undergoes zygotic meiosis, then we get the progeny, which are all peptide. So here also it shows the non-Mendelian pattern of inheritance. Suppressive peptides contain mitochondrial DNA, which is greatly altered in base composition compared to the wild mitochondrial DNA. Hence, these suppressive peptides contain mutated mitochondrial DNA, while the normal ones contain the normal mitochondrial DNA, which is also known as wild type. So the progeny which we get as a result of the cross between suppressive peptide and normal haploid cell are all peptide. There are two hypotheses to explain this result. Mutant mitochondrial DNA that is present in the suppressive peptide replicates more rapidly compared to the wild mitochondrial DNA in the zygote, as a result of which most of the progeny are peptides. And the second hypothesis is, in the zygote, recombination occurs between mutant and wild type mitochondrial DNA. This introduces errors into normal mitochondrial DNA, and now most of the mitochondrial DNA are also mutated, as a result of which most of the progeny or the offspring that we get are peptide. This is all for today's video. In the next video, we will discuss about maternal effect, taking the example of shell coiling in snail. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.